It will start off innocuous, soft, almost feathery foliage, followed by pretty yellow flowers that remind you of snapdragons, but then it spreads and spreads and spreads. The natives get crowded out, and soon it's just a sea of yellow flowers advancing over your land. Today we are talking about the invasive colony-forming weed, yellow toadflax. My name is Christy Gladham. I've worked in biological control for over 10 years. Welcome to the Colorado Department of Agriculture's Biological Control Series in collaboration with the USDA Western Sustainable Agricultural Research and Education Program. Yellow toadflax, Linaria vulgaris, is an invasive perennial plant brought from Eurasia in the 1600s as an ornamental for gardens and also as a medicinal folk remedy. It's known as butter and eggs for its vibrant yellow, snapdragon-like flowers with small spots of orange. Yellow toadflax has since escaped cultivation and now invades forests, woodlands, riparian areas, and rangelands, especially in drought-stricken or disturbed areas. Yellow toadflax begins growing in very early spring from either seeds or the large spreading root system. This is a perennial species, meaning the root system will persist from year to year, sending up shoots each growing season. The roots are rhizomatous, meaning they creep through the ground and can send up shoots over a wide area. Stems grow 1 to 3 feet in height with lance-shaped, waxy, blue-green leaves that attach singly to the main stem. Stems are typically a bit woody near the root crown. Each root system can produce over 100 shoots during a growing season. Flowering occurs from May until August. The flowers are bright yellow with 2 lips that form a mouth and an orange bearded or hairy throat. The flowers have a long spur hanging straight down that differentiates them from snapdragons. Multiple flowers are produced in a spike-like raceme or a vertical cluster. Round seed pods form that contain many seeds. A single plant may produce over 20,000 seeds that can then be wind-borne and start new colonies far away. Seeds may be viable for up to 10 years in the soil, but don't have a very high germination rate. Only about 10% end up growing. The stems will begin senescing in October and will dry out, but usually remain attached to the basal root through winter and sometimes for a year or more. Yellow toadflax mainly spreads through its creeping root system. These root systems can create large, spreading monocultures. Yellow toadflax comes up very early in the season, giving it a head start compared to other native vegetation. Large colonies of this plant can outcompete shallow-rooted perennials for water or crowd out annual plants from the very dense above-ground growth. Toadflax contains high levels of glucoside compounds, which are mildly poisonous in large quantities, especially to cattle. Animals will typically avoid these plants, and this can reduce forage significantly. So how do we control this plant? The leaves have a very thick, waxy cuticle that can make application of chemical control particularly difficult. When tillage or cultivation is used, root fragments could spread from the machinery and can propagate into new infestations. Intensive and continuous cultivation can reduce yellow toadflax, but will also allow for ideal invasion pathways of other weeds. Hand pulling may work if the entire root system can be removed and disposed of properly. Grazing is not typically recommended due to the mild toxicity of the plant. If grazing is used, corralling of 6 days for cattle and 11 days for sheep is required to allow any viable seeds to pass through the digestive tracts between contaminated and uncontaminated fields. Mowing yellow toadflax is considered ineffective. If eradication of small, discrete patches of yellow toadflax is desired, biological control will not be the right strategy to employ. In those cases, herbicide application or hand pulling of vegetation could be the right choice. However, if you have large, extensive patches of toadflax, and the goal is to reduce the density of the plants on a longer time scale, say 3 to 5 years, or you'd like to reduce invasion potential for neighboring properties, or to reduce disturbance of the land as much as possible, then biological control could be the right choice. As always, before considering collecting biological control agents, please ensure you have the proper permission or permitting to do so, and ensure the biological controls are permitted and allowed in your area. Movement of biological controls across state lines is permitted by the USDA APHIS PPQ and subject to rules and regulations. There are around 7 biological controls for yellow toadflax, many of which were accidentally released in the early 1900s and are not considered approved. They are not typically collected, nor are they allowed for purposeful release. The best approved agent, yellow toadflax stem boring weevil, Mecinus janthinis, was first approved for release within the US in 1996. Adults are cylindrical, black with a blue-green tint, and are around 4 to 5 millimeters or the size of a grain of rice. They can be found emerging from stems, typically beginning in June. The newly emerged adults will mate and females will begin laying eggs. They may continue to lay eggs into July. The eggs are found singly in little chewed holes on the stems. When the eggs hatch, the larvae will begin eating their way through the stem as they develop. They will pupate in the stem, becoming adults by fall, but they stay inside of the stems to hibernate and will not emerge until the following spring. The adult weevils will chew their way out through the stem, leaving a characteristic round hole in any of the previous year's growth. While a female may lay only one egg per stem, multiple females may use the same stem, resulting in multiple weevils developing within each plant. There's only one generation per year, which makes these weevils univoltine. Collection of these weevils is relatively straightforward and can be performed in one of two ways: either field collection of newly emerged adults or stem collections containing hibernating adults. Scouting sites to collect from will occur in June, sometimes into July. Large yellow toadflax infestations that are not sprayed, mowed, or otherwise disturbed are best. A population of weevils will be collectible when you see multiple weevils per plant. In fall, you can break open stems and find multiple overwintering adults in each stem. I would say around 5 to 10 weevils per plant or stem should be seen for a good collection site. Usually when I find a good site, the weevils are obvious on plants and I can begin field collections right away, or take GPS coordinates for a collection in the fall to release weevils the following year. For summer field collection, you will want a cloth insect sweep net, a insect collection container with plenty of yellow toadflax foliage, and a cooler with ice packs. Try to collect on sunny, clear days. Don't collect on wet foliage, as a wet insect net will suffocate and damage insects. Hold the insect net with the opening at 45 to 90 degrees and the opening close to the middle or base of the toadflax, depending on the plant height. Use a back and forth sweeping motion to firmly sweep the majority of the toadflax foliage, keeping the opening of the net facing the foliage. The weevils will be startled and should fall off into the net. You should be getting at least a few weevils per sweep, and you can make your way methodically throughout the entire patch. Periodically tap or shake down the net to concentrate the weevils towards the bottom of the net, as they will try to climb up and out. Don't over-collect, as too many insects in a net can suffocate, overheat, or get damaged. Depending on the size of your collection, you will want to periodically empty your insect net into the large collection container. For a smaller collection, you may only need to do this once. To empty your insect net, tap down the net to concentrate the insects near the bottom of the net. Grab around the outside of the net with your free hand to close it off right above where the insects are concentrated. Using the hand that is closing off the net, invert the net over or slightly inside the collection container. Push your hand through as you release your grip to get the insects into the collection container. You may give your hand a little wiggle inside the inverted net to knock some stragglers off. From here, you could place the collection container into a cooler with ice packs or into a refrigerator for a little while before sorting out the weevils from any bycatch. Cooling the weevils will slow them down and make them a bit easier to handle. To sort out releases of insects, you will want release containers with yellow toadflax foliage, clicker counter, a sorting tray, and an aspirator or forceps. After letting the collection cool down a bit, place weevils in a large tray for sorting, and use either an aspirator or forceps to collect 200 weevils per release container. The bycatch can then be released back into the field or discarded. The sorted collection cups can be stored in a refrigerator for up to 1 week, but releasing them as quickly as possible into the field is better for the health of the insects. For fall stem collections, you will want clippers and paper grocery bags or cardboard boxes to store stems. You can head out into the field after the first hard frost and when the stems have fully senesced or dried out. The yellow toadflax stems can be clipped near the soil level, then stored for a couple of months in paper bags or cardboard boxes in a cold and stable environment. A walk-in cooler, refrigerator, or root cellar could all work well. You do not want the stems to experience highly fluctuating temperatures or temperatures too far below freezing. You do want to ensure the stems stay fairly cold. You also need to be aware of relative humidity and ensure the stems do not mold during storage. You can break open one or a few senesced stems to confirm weevil presence and number of weevils per stem at the collection site. That will let you estimate the number of stems to clip and store over winter for the number of releases you plan the following spring. There are a couple benefits to this method of collection. First, you'll increase weevil survivorship and be able to maintain or increase weevil numbers more quickly. Field studies have shown that weevils overwintering in field conditions can have an over 50% mortality rate from exposure to multiple freezes. The second benefit is that you will prevent herbivory that can reduce overwintering weevil populations. Mice especially are prone to eating overwintering insects found inside of plant tissue. To prepare your spring releases from your fall collected stems, you can either place the stems directly in the field around mid-May and let the weevils emerge naturally. You won't know exactly how many weevils you are releasing, but it can be very easy and very effective to release this way. For more precise and controlled releases, you can bring the stems out to room temperature in early to mid-May, let the weevils emerge and collect them as they do. You'll want to place the stems in a sealed container, typically with mesh sides so the weevils don't escape, but there is plenty of air movement. Insect cages can be bought for relatively cheap. Once brought to room temperature, the weevils may begin emerging right away, and nearly all should have emerged within about 1 week. You can periodically check on the stems throughout the day and aspirate or use forceps to collect any emerged weevils. Place 200 weevils per release container with plenty of food. The second method is best for getting precise numbers and completely excluding the chance of transferring parasitoids from one field site to another, but it is a bit more labor intensive. A release of 200 adult insects is usually recommended per half to 1 acre of infestation, though a higher release rate may help you see a faster decline in toadflax populations. Releasing the weevils is incredibly straightforward. Prepared releases in cups can be stored for up to 1 week in a refrigerator, but releasing as close to the collection time is better for the health of the insects. Take your release cups into the patch of yellow toadflax where you would like to introduce weevils. Usually mid-morning on a sunny day is the best time for release as the insects will be more active, but any time should be okay. Don't release in very heavy rain, but if it rains after release, it is okay. The insects will be fine. Open the container and tip the weevils out into the toadflax all in one place. They will disperse themselves throughout the patch, so you do not need to scatter them. If the weevils are clinging inside of the cup, you can take a small bit of grass or soft foliage and gently brush them out of the cup. Weevils have a startle response which can cause them to play dead. It can be very convincing, but they should begin moving about in a couple of minutes. I like to take a photo of the weed patch for future reference, a GPS point, the number and species of weevil released, and the date of release for my records. Weevils will easily be seen when in high numbers in the spring and early summer. There will be very characteristic holes chewed through the leaves by the adults, and with more careful observation, egg deposition holes can be viewed on the stems. Emergence holes on the previous year's stems should be obvious, and of course those stems may also be broken open, like we've previously discussed, to determine if adult weevils are overwintering or old pupation galleries can be seen. Mecinus janthinis has been very successful for use against yellow toadflax in specific areas, but it does not always establish or persist well where it is released. This can be a bit of a hit or miss biocontrol agent. When it works great, but it just doesn't always work. Nonetheless, Mecinus janthinis can still be utilized while the search for better biological control agents continues. Speaking of which, there is a relatively new agent that was approved for release in 2018. Rhinusa pillosa, a stem-galling weevil. These weevils are roughly 4 millimeters long, or about the size of a sunflower kernel. They are black and very fuzzy. Their life cycle is pretty similar to Mecinus janthinis. The adults emerge from overwintering in March to May, right as spring growth of the toadflax begins. The weevils will feed, mate, and the females will begin laying eggs within the stems of the plant. They chew a small oviposition canal to deposit around 3 to 5 eggs, and they will excrete a fluid that covers and protects the eggs but also turns blue. This makes the eggs a bit easier to spot and identify. As the eggs hatch and the larvae begin developing inside the stem, their biology interacts with the plant to form a gall, or a deformation of plant tissue that in this case looks like the stem swallowed a ball. The gall weevils don't stay too long inside these galls, usually emerging as adults only 50 to 60 days after hatching from an egg. The new adults will then find places within the soil to estivate, a kind of summer hibernation, coming out occasionally to feed on yellow toadflax leaves. These adults will overwinter in the soil and emerge the following spring to begin the cycle again. Gall formation will be fairly obvious and can be observable within 48 hours after egg deposition. The adult emergence holes from the galls should be readily seen within 2 months of eggs being laid and galls forming. These galls may act as energy sinks from the plant root system, taking energy out of the roots for their growth and formation. The galls may interfere with the transport of nutrients or water to the upper part of the stems and can help prevent the plants from flowering or setting seed. We are not sure how well these weevils will establish or spread in Colorado, or how much of an impact they might have on yellow toadflax. It is a promising new agent that may have additive effects to other agents already established for yellow toadflax. If you find these agents, please report them in EdMaps. Damage directly on plants from the release of weevils may be seen within only a couple of weeks from release, either through feeding damage or gall formation. However, you want to see a new generation of weevils the summer after release to determine if the yellow toadflax weevils have established. Yellow toadflax weevils will decrease the plant's ability to flower and set seed, and may also shorten the lifespan of individual plants. Yellow toadflax may continue to come up from the seeds that have been previously deposited in the soil. The seed bank may last up to 10 years, so it can take a few years, usually 3 to 5, to see a definite decline in yellow toadflax abundance. Weevils alone will not eradicate yellow toadflax. Eradication of yellow toadflax from your land will take an integrated approach. Not all control methods will be compatible with either the stem mining or stem galling weevils. Herbicide in late fall could be a good integrated management, as the stems containing weevils should have senesced and will not translocate the herbicide into the plant material they inhabit. Herbicide should not be applied on plants that have active weevils, as not only will it destroy their food source and larval habitat, but the surfactants could kill the insects directly. If plants are to be hand-pulled, removing non-galled plants in early spring before stem mining weevil adults have emerged from the previous year's stems would be the best timing. Mowing, tillage, or cultivation would not be compatible with these weevils. Reducing disturbance and encouraging competition of native or desirable species will help with restoration of invaded landscapes. I hope this video has given you the information, methods, and confidence to identify, collect, release, and manage yellow toadflax weevils. In the video description, there will be a list of references and sources if you would like to read more about these helpful insects and how you can manage yellow toadflax. Thank you for joining me today.